Well, my name is Jim Handa. I am the chief of the retina division here at Wilmer. The task I've been asked to do is give a short presentation of really basically why I chose to go into ophthalmology and to become a retina specialist. And just as a plug, why you guys should be interested in doing the same. Um, so um, Really my journey first to medical school was really, I guess you have to take advantage of the problems that you encounter in life. And I was playing soccer in college and I blew my knee out beginning of my junior year, which required surgery. And the whole process of tearing my lateral meniscus and having a meniscectomy made me interested in going into orthopedic surgery. But when I hit orthopedic surgery during my rotations, I just didn't fit in. And since it's being recorded, I can't really say exactly why, but I just didn't fit the phenotype. And through all my rotations, I really sort of fell in love with ophthalmology a little bit late. And I think some of the key reasons why I went into ophthalmology were basically the importance of vision. If you look at patient surveys of what they're most afraid of, it's losing vision, losing life from cancer, and heart disease are the top three. I thought that the elegance of microsurgery was just really phenomenal. All of the work we do is below tactile sensation, so you have to learn visual cues and other things to make these minute maneuvers under the operating microscope. And it gave me the opportunity to have a really diverse patient population. I personally deal with prematurely born babies, but also centenarians, people who are in their hundreds. And I found that ophthalmology has an intellectual approach similar to neurology. And I'm basically a nerd, so it appealed to me. But there's therapy, and you can really make a difference in people's lives with the surgery, and that was appealing to me. And I love the surgery, but I didn't have to deal with some of the strong personalities that you'll see on your surgical rotations. And finally, it gave me the opportunity to do research as well as have an active surgical and clinical practice. So why retina? So I thought sometimes a picture's worth 1,000 words, and I thought I would show a video, and you can see, here's the video. This is of a vitrectomy, and you can see that this is a case of an intraocular foreign body. So a piece of metal was shot into the eye, and we were tasked to remove it. And you can see we make 3 incisions. These are 25 gauge in size, so they're smaller than most IVs. And this here is where there's an infusion line to keep the eye pumped up, and the other 2 are the working ports. And you're going to see that that we're making an incision into the sclera with a super sharp blade, because that's where we're going to take the foreign body out. And again, everything's a little bit magnified just because it's under the operating microscope. You can see some of the fluid starting to egress, and it's plain fluid because we've done a vitrectomy up to this point. We want to remove the vitreous gel because it's gooey, and if we pull the foreign body out with the vitreous gel, we'll take the retina with us. And so here, I want to draw your attention up here, where you can see the forceps that grabs the foreign body and bringing it out of the eye. And then you can see it coming out there, the foreign body. And left in there, a piece of metal, the toxicity from the metal could potentially cause a retinal degeneration and blindness. So why academic retina? I think we're always influenced by our mentors. And I certainly got inspiration from one of my mentors during my fellowship when I was at Duke. I had the opportunity to train under Robert Mockemer, who's an extraordinarily gifted scientist and creative thinker. He was the inventor of vitrectomy. And I show these pictures. These are pictures he gave me. This was when he was developing vitrectomy, and you can see him actually working in his garage on the weekends to figure out the task he thought of removing vitreous gel from an eye was similar to taking out an uncooked egg white from an egg without breaking the yolk. And so he developed an instrument, and you can see the egg shell, and the prototype was run on 3D batteries. And there was actually a shaft here that was hollow that aspirated the vitreous or the egg white. And there was the original design was sort of like a screwdriver that would sort of twirl the vitreous, and there was a guillotine cutter like we have now, but it would sort of twirl the vitreous up into the shaft of the device, similar to the way you sort of twirl noodles around your fork when you're eating spaghetti. So that was really inspiration, and just the whole process of how he developed the procedure was something that I thought would be very interesting from just a general research. My interest has been more in the laboratory, so most of my work is in the lab. So I run a research lab. I have for the past 30 years or so, and I've been interested in how smoking contributes to age-related macular degeneration, development, and progression. So I'm interested in how the eye transitions from chronological aging to early disease. And since smoking is the #1 environmental risk factor, I focused on that. And one of the models that we've developed is by exposing laboratory mice to smoking over 6 months, and they develop an AMD type phenotype. So one of my reputations, I'm known as the smoking mouse guy. So why a clinician scientist? So for me, I think the perspective is you really get to know a disease inside and out. And from a day-to-day perspective, there's this bi-directional link between clinicians and scientists. And for me personally, I think that as a clinician, I can provide direction to basic scientists who can then target some of their scientific approaches to better understand disease pathophysiology. And all of that information is then really translated into all the textbooks that we read. But I can also take ideas from patients to the lab, and I've had some interesting patients who have generated research ideas for grants. And the research, I think, has taught me to listen to my patients differently and to think about their problem from a different perspective. So some of the routine, boring patients, I think about what they have to say a little bit differently than others. So each patient is a little bit more interesting to me. And I think from a global perspective, some of the work I do involves being involved in clinical trials. And so you have the ability to get involved in testing new treatments. So what is my day like? So, I think one of the things that struck me was a colleague of mine, when he turned 50, he was really pretty burnt out because he sort of did the same thing day in and day out. And for me, every day is different. And that, I think, is what keeps my life very invigorating and refreshing. So on Monday, I start my day with our retina teaching conference, where we go over interesting medical and surgical cases with the other faculty, residents, and fellows who attend. And then it's research in the morning, and then in the afternoon, I'll see patients. Tuesday is my OR day, so I'll typically operate for probably 2/3 of the day, and then the rest of the day, I sort of clean up with research, things that I need to do. Wednesdays, I see my post-ops early, and then I have my lab meeting, followed by a day of research. Thursday, I start out with grand rounds, so we get to see a lot of different interesting cases that are not confined to retina, so I keep fresh on my comprehensive general ophthalmology background. And then it's research for the rest of the morning, and then I have clinic in the afternoon. And typically Fridays are for research. I also have attend the clinical trials meeting that my colleague Peter Campachero runs. He currently spearheads about 30 different clinical trials going on just in our retina division. And I think one of the things that I think is really interesting about retina in particular, but so much in ophthalmology, is that the landscape is changing on a daily basis. And so the future is very bright. I just show a picture. I had the opportunity to be involved in the research, the clinical trial for the Argus 2 vision chip implant that was FDA approved for patients with advanced retinitis pigmentosa. And here you can see how we have this array that sits on the retina. These are electrodes that will stimulate the retina from a digital video signal that's converted to electrical impulses that a nerve can understand. And that information gets sent in real time to the brain. So there's a very crude visual image performed, but some patients have gotten to the 2800 level. And it was very rewarding to be involved in that. There are better devices that are being developed and being tested as we speak now. On the other hand, OCT angiography and a number of imaging modalities have emerged, which really deepen our understanding of retinal diseases. And here's a OCT angiography image where you can just make images from flow of blood cells through the different retinal vasculature in here in the macula. And the future is now. So here's a video image of the RGX-314 gene therapy trial. So an AAV virus is injected very carefully subretinally with a 42 gauge needle to make a couple of blebs that will infect the retinal pigment epithelium and other cells within the retina. They secrete basically Lucentis, or an antibody fragment that neutralizes vascular endothelial growth factor. So this is a sort of a permanent fix for to try to stop having to give the monthly anti-VEGF injections for wet macular degeneration and other diseases. But there's also gene therapy for RPE65, which is FDA approved for retinal degenerations, and others are coming down the road. So again, as I say, the prospects are very bright for innovative medical, surgical, and imaging approaches to ophthalmology. The other thing that I think an academic life has brought me was the ability to, at times, control my life. So for me, I've been able to achieve what I think is an optimal work-life balance to bring happiness. And I think it's more important to be happy than successful. I know many successful people who are unhappy. And what my wife and I did was we made it a priority to emphasize raising our kids. And both of my kids got involved in sports. And when my daughter was playing field hockey, competitive field hockey, and for her high school team, I was the one dad who went to every one of her games. And my son was involved in cross country. I was able to go to his meets, and I think I had the flexibility to rearrange my day so that I could go to what their activities and events, and then I might just shift my day and work a little bit at a different time of the day. So I think the flexibility is what allowed me to be actively involved raising my children, which was obviously very important to me. So my last slide is really to provide some advice to first and second year medical students who are interested in going into ophthalmology. And I think my approach is hopefully a little bit different, and hopefully it can change your perspective on how you view doing your things in medical school. I think you should appreciate how lucky you are to be in medical school. So enjoy the journey. And I think back to what I did. The last course I took in medical school was forensic pathology, because it was something that I would never do. And I was able to go to an autopsy, see autopsies. I was able to go to court to see how the testimony from forensic pathologists could turn a murder trial. And I was able to go out to the field to see how pathologists will evaluate a crime scene, something that you can't do. So take advantage of the diversity of courses that you can take in medical school. And I think it's important to stay intellectually curious. Always question whatever course that you're in or whatever you're doing. You'll just learn more, and it becomes more fun. I think pragmatically, I think it's really valuable to do a research project with the intention of being a first author on a publication. And that's even if you don't want to do research. I think if you if you get involved in research so that you're a first author on a peer-reviewed publication, it elevates the commitment that you have to make, and it'll give you greater insights into what makes a good paper, what makes a bad paper, and it will make you a better ophthalmologist because Learning is lifelong, and so we have to stay on top of the literature, no matter if you're in practice or in academics. The other practical component is you get to know your mentor and other ophthalmologists in a meaningful way. So it's not only enriching, but it's pragmatic. You can get a worthwhile letter of support as you apply. And then finally, just be outstanding. There's no question ophthalmology is competitive, so you do have to do well in your coursework. But the perspective I hope you have is to immerse yourself in the material. If you really enjoy the material you're learning, the grade will come. And finally, showcase your talent. I personally, when I look at applicants, I look for outstanding. So if somebody has been an outstanding athlete in college, I know the dedication it takes to be successful. Or if someone is a really outstanding pianist or violin player at a high level in the orchestra, I think you should showcase that, because to be an expert, you need 10,000 years, or 10,000 hours, and it takes dedication. There are bumps in the road, and learning how to overcome the adversity to become outstanding is just going to make you stronger. So, so be outstanding and showcase your talent. I think that's basically what I have to say, and I'm more than happy to answer any questions that people might have.
Great. So we have a list of questions that were submitted from our students on the form right now, but I want to open up the floor and see if anyone wants to put a question in the chat, or you can direct message me. But I can go ahead and start with the questions from the form if that's okay, Dr. Handa.
Sure.
All right. Excellent. So thank you for that wonderful talk. Let's see. Our first question is, what are the global health opportunities available for retina specialists?
Limitless, actually. I think, if you think of major diseases, I think with third world countries becoming westernized, diabetes is probably the major disease to think about. It's really becoming almost an endemic in many developing countries. It's interesting. One of the things I didn't mention is for the past 30 years, I've actually provided goodwill mission care to a small island in Micronesia called Palau, which is about 600 miles east of the Philippines. And it started by a friend of mine giving a lecture on diabetic retinopathy to primary care specialists. And that was right around the time that lasers went from water-cooled to diode technology, which meant that the lasers used to be the size of a large closet, and now they're the size of something that you can fit in a carry-on suitcase. And so his last slide was a picture of the diode laser with a suitcase, and he said he could go anywhere. And there happened to be a primary care doc who was stationed. He was a U.S. Public Health Service doctor who was stationed in Palau. And he came up and said, I've got all these diabetics, but no one to take care of them. Would you be willing to come? And Palau, if you don't know, Palau happens to be one of the premier scuba diving places to go in the world. And I was a scuba buddy of this person, Mark Whelan, who we were co-residents together. And so he called me up, and we've been going over every year. So we typically go over for a couple of weeks. We see patients. So we'll see probably close to 1,000 patients. We'll laser patients, we'll give injections, we'll treat anything. We used to do cataract surgery along the way. Very rewarding. We've learned how to speak a little bit of Palauan to get by. And then we take 3 or 4 days and go scuba diving. So it's a win-win. We estimate we save the government approximately $250,000 each time we go over. When you consider to get the care, they have to fly to Honolulu and get care. So if you include travel expenses, it's really very expensive. So I think that's an example of the kind of things you can do with the emergence of telemedicine and non-mid-radic fundus cameras, where you can take pictures of retinas. I think the expansion of being able to understand diabetic retinopathy in particular is going to be something that is going to take hold globally.
Great, thank you. Just received a question in the chat asking if you could speak a little bit more on the difference between medical and surgical retina in terms of pathology seen, procedures, day.
Yeah, so I think that's really one of the really the selling points of ophthalmology, because you can be a medical specialist only or a surgical. So the difference in medical retina is that medical retina specialists tend to focus on um, age-related macular degeneration, inherited retinal dystrophies, for the most part, like retinitis pigmentosa, um, diabetes, up to the surgical stage. So they'll take care of anything in the office. They will do lasers, they'll give injections into the eye, but they will fall short of going to the operating room. Surgical retina is really all about fixing retinal detachments, and that the standard rhegmatogenous retinal detachment, where there's a tear in the retina, and putting the retina back in place through the different techniques we have. As you saw from the video, there's um, intraocular foreign bodies or trauma that will fix retinas from that perspective. Will drain hemorrhage underneath the retina after trauma or other surgical procedures, and advanced diabetic retinopathy changes. Premature babies develop retinopathy of prematurity, which is a very complex type of retinal detachment. Will fix that. So the practice is geared where you typically will manage some medical retina patients, but when they transition to a surgical disease, then you take over. I think most of the medical retina people are very, very heavy into research. And I think the perception is that they want to focus on the research that they do and not be interrupted by surgical emergencies, things like that. So I think that's really the main distinction. But I think if you have, I think I like the versatility of surgical retina, and I'm clearly biased because that's what I do, but it gives you the opportunity to take care of a patient from the beginning to the end and not hand off the patient. So I like that continuity of taking care of a patient in that respect.
Great. Our next question is, how much of retina work involves trauma and being on call?
Well, it really depends on the institution you're at. I leave a very cherished life here at Wilmer, because we have in the retina division we have 18 faculty. So we typically take call every for a week at a time. So I take call every 18 weeks. We also have the benefit of having outstanding retina fellows who are in training, so they're the first line of defense. So when I'm on call, the only time I basically get called is to go in to do surgery. So life's not so bad doing that. I have to admit, as I get older, doing a case at 1 a.m. doesn't have a lot of appeal to me anymore, but exciting nonetheless once you get there. It really does depend on the type of call. It depends if you're in practice. It also depends on the size of your practice and the kind of practice that you want. So there definitely can be practices where you can avoid being on call and just kind of do your own thing. I think there's flexibility in how you construct the kind of practice that you want.
Are there any specific experiences or reasons that you feel majorly influenced your decision to subspecialize in retina?
Well, I think the overall complexity of the retina was really a driving factor. And what I've told people who actually ask me in private, to be honest with you, when I started my residency, I thought I was going to do glaucoma. And the reason is I thought that understanding how glaucoma develops was a complex thing, and it would be fascinating to really understand that. And for me, the surgery didn't appeal all that much to me. And the first time I saw a vitrectomy, when you put the lens on the eye and see this comprehensive view of the retina, it's a strikingly beautiful organ. And frankly, that turned the light on. It's hard to say that convincingly when you do a fellowship, but that was really a turning event for me. And I think it really forced me to then start to read about retinal diseases a little bit more and think about how I wanted to construct my career. And I thought that there was such a diversity of different types of diseases and approaches to management that it would be hard to get bored in that kind of practice.
So one student is asking if you have any advice on how to make of conferences like ARVO as a medical student, like what kinds of things you can do, what you should be.
So you froze a little bit. So the question was something about ARVO. Can you repeat that?
Yes. So the student's asking if you have any advice on how to make the most of conferences. So when they go to conferences, what are some things that they should be doing?
Okay. Yeah, I mean, I think if you're interested, going to ARVO or the Academy are fantastic ways to get immersed in it. So first you have to have a project so that you can submit an ARVO, an abstract. Just inside information, ARVO accepts about 90 or 95% of abstracts submitted. So you have a really good chance of getting something accepted. When you go, I think it's always helpful to look at the program before you go to try to figure out what are the interesting topics that appeal to you. But at the beginning, you should try to sample things in all of the specialties of ophthalmology, from cornea to plastics to retina to glaucoma to neuro-ophthalmology, et cetera, so that you can get a really strong understanding. I think spending time in the topic of whatever your research project is really great. So I think while going to some of the lectures are great, I think the most invigorating conversations that you can have are by going to the poster sessions. And whether it's a topic that just you're curious about, or if it's a topic similar to the research that you're doing, you can go and have conversations with people who are at your level, but also really very, very senior established researchers. And in an informal environment like that, people are always very receptive to having a conversation. And I think you can meet people. I have to admit, when I was a medical student, that's when I met Peter Campicero, who's one of my colleagues in the retina division. And I was just curious about his research. And he was sat down, and he's not a man of a lot of words, but I sat and talked to him for about 30 minutes. He probably doesn't remember that conversation at ARVO one year, but very inspiring. And so I think people are very, very approachable in those kind of informal situations. And the other places, when you're not in COVID, is there always are places where people tend to congregate. And so having a beer with a bunch of people is a great way to really immerse yourself in it. So just experience everything.
So we have someone asking about advice that you have for students who didn't have exposure to ophthalmology until their third year, which is very understandable because many students aren't really exposed to it in early years. And how can they make themselves outstanding in ophthalmology at this point in their training when they're already in their third year?
Well, there's two ways to do it, and I don't think there's a right or a wrong way. Obviously, one way is to consider taking a gap year. Um, the gap year can certainly allow you to pivot so that you can get some experience, whether it's working in a clinic or getting exposure to ophthalmology to make sure that's what you want to do, or to do a research project. The research project, if you do a research project, it's probably better to do a clinically oriented project, because you can have a small enough project that you can get a publishable unit out there. Because if you take a year off and you start applying, time passes very quickly. And so you want to make sure that you demonstrate some productivity, that you're moving forward in your career. The other way of doing it is, again, if you're doing well in medical school and you have other aspects of your application that demonstrate that your talent, that where you can being open and honest, saying, look, I'm new to ophthalmology. I discovered it late because, or whatever. I know this last year we looked at an application from a very small medical school of a student who decided late and didn't have any exposure to ophthalmology, but managed to go away for a month to an institution that had a big program just to get exposure. And just in the personal statement describing the inaccessible, the lack of access to an ophthalmology program, but this is what happened in developing an idea late. That combined with, here are the strengths of what I represent, and I have basically lots of potential. I think that can be an equally satisfactory and competitive way to do it. So for me personally, I don't like to see someone become a professional student going to 10 or 12 different ophthalmology programs on a rotation. I think you want to do one or two just to really convince yourself that you want to do it. But I don't think you want to project yourself as a professional ophthalmology student to try to impress everybody.
All right, that's good to know. Thanks, Dr. Handa. We have another question that's asking, how can it be possible to learn more about or get involved in future developments like the ARGIS-2 and newer developments in the OCT, clinical trials, technological innovations? Is it better to focus on academics, matching, and that kind of stuff, return to the future, or how could a medical student involved early in their career?
Yeah, I mean, I think so. There obviously are different. So, for instance, with the ARGIS-2, it's interesting. The ARGIS-2, the company actually went bankrupt, and it's an interesting lesson, I think, in making sure that you have all of your data correct. And what I mean by that was the science behind the development of the ARGIS-2 was spot on. I think from a personal perspective, I think where the company ran into problems to go bankrupt is they overestimated the number of patients who would be eligible for having the ARGIS-2 place. So in the United States, you have to have light perception vision or no light perception vision. And the number of patients who have retinitis pigmentosa and have that and who want to get the implant was projected to be 100 a year, and it took there were 100 over, I think, 4 years. So the recruitment was way behind, and so that put them financially in a bind, and so they eventually went belly up. There are other ones coming down the road, and I think there still is a lot of research coming out from the ARGIS-2, and so being involved in a program where you can sort of retrospectively analyze some of the data that's been accumulated would be helpful. I think with any of these kind of vision chip implants, for a student to be involved on any kind of meaningful basis would have to be more analyzing data that's already been collected. So it's more retrospective rather than prospectively being involved. To become more prospectively involved, it would require you to take a year or two off at a minimum in order to have a meaningful experience, which can be a great experience, but it for people who want to sort of get on with their life, that may not be quite so appropriate. On the other hand, getting involved in imaging, I think is a really fruitful way to go. So, whether it's OCT, standard OCT, or OCT angiography, or any other kind of imaging, I think the data sets can present themselves very rapidly. And if you can come up with an interesting question on a disease, it opens up an opportunity to really learn how to interpret these images. And I think, I think the benefit is that you can become an expert in analyzing these images, and more so than people who are going to interview for ophthalmology residencies. And that's another advantage of immersing yourself in these kind of projects. When you go to interview for residency, the dynamic changes. You go from being asked questions like, why did you go into ophthalmology, to tell me about your research. You tell them, and then you educate the people who interview you. So you're kind of reversing roles. And I think that's really an advantage to sort of make a really solid impression when you're going through your interviews. So I think of the two examples that were raised on getting involved in research, I think the imaging side offers a really quick way to make some headway into really understanding, becoming an expert, and doing some nice research.
Great. Our next question asks, and I know you touched on this a little bit by talking about going to another institution, but the student is asking what advice you have for students who don't have an ophthalmology residency program or research opportunities at their own institution? And also, how much weight does previous research experience publications have in the residency application process?
Yeah, so the first part of the question, if you don't have much of a program, I think that's where it really is helpful for you to go apply and take an away rotation. And at the same time, if you can establish enough of a relationship with somebody to do a research project, I think that'll help you out quite a bit. And it could be something as simple as a case report. So if there's an interesting case, learn how to write it up, become an expert on that particular disease. It's fun as well. So I think that's the way to go if you don't have a program at your medical school, because you want to, I think from a practical standpoint, it's always helpful to demonstrate the experience, because otherwise people will ask you, well, how do you know you want to go into ophthalmology? And also, I think if you can, as you select recommendation letters, the faculty, I think it's always most important to have someone write a strong letter for you who knows you, so they can write something meaningful. I think that's better than having a big name who doesn't know you, because there's just going to be this generic letter, and they're just going to recite your CV, and that's not very helpful from my perspective. And it's always nice to have someone in ophthalmology write a letter for you. So if you don't have a program, going to a program so that you can get a letter written in there, and that's very common. We see that very commonly, where someone say, well, they rotated with us and I was happy to write a letter. And that is very helpful, because if I know who the ophthalmologist is, it's a little bit, it's obviously helpful. So I think for those circumstances, it really does help, and it's almost essential, I think. What was the second part of the question? I'm sorry.
That's all right. The second part was asking about the weight of research in the application process.
Yeah, I think the weight of research is for the most part secondary, because I think your academic performance is more important, and then the dean's letter will provide some perspective on how you rank in your class, always in sort of a cryptic way, so you're never completely certain. And then yeah, and then I think. It also depends on the institution. So, putting it out there, Johns Hopkins is known as a research heavy institution. So if you're interested in coming to Wilmer, I think research is looked at more seriously than other institutions. If you were interested in Bascom Palmer, that's a more clinical institution. Wills is a more clinical institution, despite the fact that I train there. Uh, Research is they're interested in research, but I don't think it has quite the priority that a Wilmer has.
All
Oh,
right, topic, go ahead.
Maya, sorry. I think there's just one more aspect of that question, and I think it was how much weight does previous research before medical school, so like research and publications before medical, like done before medical school, have in the residency
I
application
see.
process?
Yeah, I think me personally, it's very helpful, because when I see the research that people have done, candidates, applicants have done, I'm curious. I want to see what they do. And I like to see what role they played, right? You can tell a little bit by the order and authorship. But I will not uncommonly, if the title is interesting, I'll go to PubMed and I'll look it up and see what it's about, because I want to see if it's interesting or not. I mean, I think I'm intellectually curious. I want to know what people do, and I like to ask the candidates about that. And sometimes they're surprised that I'm going to talk about something they did when they were an undergrad. So I think it's meaningful. I definitely do. And it's also, I think it's helpful if you have a nice publication like that you've made a meaningful contribution. I think it would be helpful in your application, whether it's in the personal statement or somewhere, to really kind of highlight a research project that you did previously that is important to you, because and that may help, particularly for those people who are a little bit late in deciding to go into ophthalmology. It sort of lets people know that you're intellectually curious, that you you do like the rigors of research because it's an important component to being a good doctor. And that sort of mitigates the concern that you might have that you don't have this, big publication in an ophthalmology journal.
On the topic of research, we have another question asking if there are any major differences between doing research as an MD versus an MD PhD.
I think fundamentally no. In terms of the kind of research that you do, I think for students, it really kind of depends on the project the mentor provides and how much time you have. I think in the long run, if I'm understanding the question right, I think, if I had to do it over again for what I do, I would probably would do an MD PhD program because and it may be maybe me, but because I don't have the PhD background in some fundamental biochemistry and molecular biology, which is a lot of the work that I do, if I have a problem with an assay, it takes me longer to figure it out. Whereas some of my MD PhD colleagues, because of their PhD background, it's kind of innate where they can sense where the problem and how to troubleshoot. So I have to read a little bit more. I have to talk to colleagues a little bit more to figure it out. So I think from that perspective, I think that's the distinction. I think I don't look at it overall in terms of MD versus MD PhD. I look at it in some ways in terms of the type of research that the MD or the MD PhD wants to do, and they're not they don't the degrees don't define what they want to do. And what I mean by this is in my division, we have probably the best scientist on the clinical side who MD PhD is doing really phenomenal work, and Dr. Akrit Sodi, and he's taken the time to build a research infrastructure so that at the beginning of his faculty career, he really built the foundations to do high impact research. And by that, I mean, the depth of his research, it's very, very rigorous. So it's not just one assay to prove a point, it's multiple assays to make sure that you cover the flaws of each assay so that cumulatively, as you read it, it's very comprehensive and you can't find any holes in the research. That takes a lot of time, it takes a lot of depth, but the amount of work that goes into it is substantially more than some other types of research. And that's a decision that you have to make. It's expensive, it's time consuming. You don't have as many publications as some smaller sort of unit publications. So some of that is dependent on the kind of research you want to do, but also the kind of grant support that you get to do it and the money that you have coming in. So it's all kind of tied together.
What was the work-life balance like for you when you were in residency?
For me, it was easy because I wasn't married. I didn't have children at the time. So I could be super selfish on whatever I wanted to do. I think ophthalmology residencies, particularly now that you have an 80-hour work week, right? So part of that 80 hours is when you're on call. So for me, I was able to pursue a lot of things. Like, for instance, I did triathlons. I trained for triathlons during my residency. So I'm kind of a morning person. So I would get up early and train in the morning before I'd go in and work, and then do my work, and then always some time for reading and following up on things. But still absolutely had ample time to get together with my classmates. And I think what you will all experience if you have good friendships in medical school, I think your bond with your fellow residents will be stronger because you're going to spend a lot of time. You're going to go through the aches and pains of learning ophthalmology, but the experience is more intense, and you spend a lot of time together. So as a result, there's this really nice camaraderie, and I felt in my residency that they were really some of my closest friends, and they take a bullet for me, and they were all at my wedding. So hopefully that kind of experience you'll have when you do your residencies.
Great. All right, we have time for maybe about 2 more questions if anyone wants to submit any last ones in the chat. I have one right here. asking about what the outlook is on retinal transplants for the next few years.
Yeah, that's actually one of the more common questions that many of my patients ask me. And 7 or 8 years ago, I would kind of chuckle and say, well, that's just not possible. But with the advent of IPS stem cell research, I I think that we're probably looking at 5 to 10 years away. So the ability to take IPS cells, so red blood cells, take and convert them into a pluripotential stem cell, and then we can now make retinas in a dish that actually, if you shine light on them, will actually respond like an ERG, electroretinogram signal. There are now some vascular channels that are sort of being modeled into it. And in terms of the transplant, really in retinal disease, for the most part, the main cell type that you really need to transplant are photoreceptors, right? They detect light. And there's some really pretty innovative ways of transplanting photoreceptors, but also one of my colleagues here is actually transplanting photoreceptors and retinal pigment epithelial cells. So the idea is if you can transplant both and lay down the foundation of RPE cells, they support the health of the photoreceptors. And so if they survive, they're going to keep the photoreceptors healthy. I think that technology is here. So I think the barrier that is confronting most transplant surgery is to have the transplanted cells actually synapse and integrate and transmit the light signal from their light detection into the rest of the retina to be able to then go back to for the patient or the animal to understand what the light being transmitted is. So I think the future is very bright, and I think it's pretty close to now, just because it's also one of the audacious goals of the NIH to really emphasize this kind of research. So there's lots of money going into it. There are companies that are investing heavily into it as well. So I think it's an area of big concern, because once you lose retinal cells, there's no way to get the vision back unless you replace the cells.
Great. And for our final question for today, what are some of the hardest parts of being an ophthalmologist? Or
Hardest parts of being an ophthalmologist? And then you were cut off. Was that the question?
yeah, ophthalmologist, more specifically retina.
Um, I think any kind of failure when patients go blind. Again, the impact that blindness can have on people can be profound. There are data that the anxiety and the depression of going blind is as powerful as being blind. So I think we deal with blinding diseases in retina, as opposed to some of the other specialties where you're improving vision, but you don't necessarily deal with blindness. And so I think that's a hard thing. And even something medical, such as a patient who has age-related macular degeneration, as somebody gets older and they lose enough vision that they can't drive anymore, is a significant loss of independence. And having to tell a patient that their vision is no longer good enough to be able to drive is really a seismic shift in their lifestyle. They can't go to the grocery store to get food. And so the loss of independence is pretty striking. So I think that's the hardest thing. I also take care of oncology patients. And so, patients don't typically, when they come into the office, they don't think about having a life-threatening disease. And so their blurry vision, they oftentimes think they just need to get glasses. And when you have to tell them they have a melanoma in the eye, and it's life-threatening, it needs to be treated, it's a fairly sobering thing for them. And then, of course, even though our treatment is excellent with brachytherapy, the success rate is about 83 to 85%, 5-year survival, but there are people who die. And to have to learn that a patient, despite adequate treatment, has developed metastasis and has a life expectancy of 1 to 2 years is a sobering thought. So I think those are the hardest aspects of retina.
Great. All right. Well, thank you so much, Dr. Handa, for this amazing talk that you gave us in the first half of the session, and for being so thorough in your responses to our questions. We really appreciated having you today.
Well, great. I wish everybody the best of luck. It's competitive, but it's not impossible. So hang in there and go for it. It's a great field. I have zero regrets, and I would do it all over again if I could.
Wonderful. We just put in the chat, I think Christina is getting drowned a little bit by everyone's response. If everyone could just hold off for 1 second. I know we all want to say thank you because it was an awesome talk, but Christina, if you want to chat one more time.
I'll just put it again. You don't have to hold back.
Perfect. There's the sign up right there. If you want to go ahead and click on it again, please give priority to those who emailed Dr. Loretta from the last session. Maybe wait a day or so. And then tomorrow, if there are still spots open, go ahead and put your name in. We are looking to put more spots very soon. So if you don't get a spot this time, hang tight, you will get one eventually. Again, thank you so much, Dr. Handa, for all of your time. It was a really wonderful session. I think the students really got a lot out of it. And thank you all for participating. Stay tuned for an email with the sign up link that you just received in the chat, as well as the recording of this session, and then more information for our March session.
Yeah, the March session is going to be also have a lot of information about global health in ophthalmology too. So if you're interested in that, it's with Dr. David Friedman from Mass Pioneer, who actually used to be at Hopkins.
A good friend. He'll have lots of great things to say.
Wonderful. Well, everyone have a wonderful evening. Thank you again for joining us. It's been great organizing this with all of you. We appreciate your time, and we hope to see you next month.